We all know that the spatial organization of the nucleus is very important. And not only the chromatins are organized in a specific location, there are also proteins, RNAs form different nuclear bodies that have to be at the right place. And this kind of spatial organization and also temporal organization of those molecules in the nucleus in proper function and this often is disrupted in cancer cells. Today, I want to talk about you a story where we focus on the disruption of the PML body, which you show here, the disruption in the PML body in one type of the cancer cells. And so the cancer cells is called odd cancer cells because it use an alternative lengthening telomere pathway to maintain telomere length. So telomeres, right, are the ends of the chromosome that shorten with every cell division. So when telomeres become too short, the cells trigger cell cycle arrest. So for cancer cells to keep dividing, they have to activate maintenance mechanism to put the lost telomere back. Majority of cancer cells use telomerase, which is a reverse transcriptase that uses a RNA as a template to put the lost telomere DNA back. And the odd cancer cells use a DNA repair mechanism that use a homology between the telomeres to repair the telomeres. And the molecular mechanism is still not super clear, but interestingly though, although odd cancer cells only accounts for a small percentage of cancer cells, for telomerase positive cancer cells, when they are subject to telomerase inhibitor, some of them will adopt the odd pathway to live. Again, the molecular mechanism not well understood. That is because the odd pathway is a very messy pathway that there are many phenotypes that we don't know which ones are the key ones that contributes to the origination and maintenance of this pathway. For example, showing you the chromosome spread of the non-odd cells versus odd cells, you can see the telomeres are nice and similar length for the non-odd cancer cells. In the odd cancer cells, you will see some telomeres are very, very long and others are short, right? You barely even see they have telomere DNA. So, and in addition to that, there are many, which another one of them is the mislocalization of the PML body that I mentioned at the very beginning. So normally PML bodies are not associated with telomeres in normal cells. However, in those odd cancer cells, we see colocalization of PML body, a subset of PML body with telomeres. As you should see, this is a cell line, odd cancer cell line, E2S, commonly used in the lab. And you can see the PML body colocalized with the telomeres to form what's called APBs, which is just PML bodies that are localized to the odd telomeres. Interestingly, telomeres also cluster within the APBs, and that's been proposed that APB is where the homology directed DNA repair happens because one telomere can use another telomere as a template. And this is a very unique marker that is used in diagnosis, and showing an example of in the tissues, cancer tissue section, you can see the localization of those two PML bodies with the telomeres, and very easy to spot. So the question we want to ask is, how does this kind of abnormal nuclear organization in odd cancer cells arises? And how does it contribute to the odd pathway and other cellular function in the odd cells, such as the PML body localization to telomere and the clustering of the telomeres within the PML body? So we have a collaborator, Roger Greenberg at Penn, who developed the assay where when they introduce DNA damage to telomeres, and they can induce telomere clustering, formation of APB, and clustering within APB and other odd phenotypes. When we look at the movies, and I found that the event of the telomere clustering that in this movie show that really resembles a droplet coalescence, and meaning that if we quantify the aspect ratio, we can see that which is defined by the length and the width of the object, that it decreases over time, and then reaches 1. And also, if we bleach the APBs, and you see recovery. So this kind of hints at this liquid behavior of those APBs, and this has been shown by this liquid behavior, shown that been shown when proteins undergo phase separation, where under some conditions, showing you a movie of protein, and where instead of being dissolved or forming aggregates, they form this liquid droplets or condensates, where inside they have very high concentration, outside they have a low concentration, but there's always change of material between inside and outside. And because of the interaction between the proteins is weak enough so that they can leave. And of course, they have surface tension, so they can fuse when they meet each other, and then they can round up. So, and this phenomena has been reported to undergo the underlying formation of many nuclear structures, including many nuclear bodies, right, such as even PMO body. So the hypothesis we came up with that the PMO bodies on the telomeres are just like liquid droplets. They get mislocalized to the telomeres through a DNA damage signaling, and then this leads to a local condensation of PMO body on the telomere to form APBs. And since they're liquid, so they can fuse with each other to cluster telomeres together. And also because the PMO body condensation process is a way to enrich DNA factors to create a compartment that DNA factor, repair factors can be enriched on the telomere. This is important because our telomere ends on linear DNA, and the DNA repair factors are normally excluded from that region so that the linear DNA, the telomere DNA is not recognized as a DNA damage, and which will cause chromosome fusion. So we hypothesize that once telomeres are clustered and we have enrichment of DNA repair factors, this will lead to the telomere elongation through homology-directed DNA repair. To test this hypothesis, what we want to do is to use tools to induce de novo APB formation in our cells. The reason for doing that is DNA damage signaling is a very complex process that induces a lot of modifications, and we cannot decouple the functional consequences of those from the APB formation that we proposed here. So the tools that we use are based on a modular design of photosensitive chemical dimerizer. And so we choose two small molecules that can interact with their protein, which is a TMP interacts with EGFR, and a halo commonly used halo ligand system, halo tag system. And then we simply put the linker in between. Now this become a dimerizer. It dimerizes two proteins that being fused to EGFR and halo. To make this photosensitive, we put a photo cage on it and showing here the first version we use MVOC, showing you here the chemical structure, and this collaboration with my postdoc lab and David Shanowith's lab, the Michael Lapson lab and the David Shanowith lab. And over the years, we've developed many versions of this because modular design, we can swap things around. So I showed you the version where we have MVOC as a cage. So this is called NTH, that is a cage molecule. And we also can simply put the MVOC photosensitive cage in the middle. Now it become a uncaged but cleavable, meaning that you can now release a protein from another protein. Or if the halo is anchored to a structure, now you can release the protein from a different structure. And then another one is a different cage, and where we have a coumarin that is sensitive to different wavelengths of light. And then we can then put coumarin as the cage and put MVOC as a linker. Then we can have a caged and cleavable. And we've used this to control several biological process, including organelle transport and mitosis. And recently, we've also optimized the linker to make it more permeable to the cell, and we've used to control phase separation, where we fuse the halo to a oligomer to act as a seed for phase separation. And then we fuse the EGFR to a phase-separated protein. And in this case, is the C-terminus of HNRAPA1 protein, and that is disordered, is known to be able to self-interact and then drive phase separation. And you can see that when you add the dimerizer, you start to see droplets forming in the nucleus, but as well as in the cytoplasm where the proteins localized with different concentration. So for come back to this project of APB project, where we want to use the tools where we're going to use recruiter protein to the telomeres to form de novo APBs. And instead of recruiting PML protein, we want to mimic the signaling after DNA damage response that will recruit PML protein. So from the literature, we know that the post-translational modifications of stimulation, which is the addition of the small ubiquitin-like modifier to the telomere proteins, is required for the formation of APB, meaning that if you inhibit, you have less APBs. Also, when we look at proteins that are listed within APB, we find they either contain sumo sites or contain a SIM, which is sumo interaction motif. And sumo SIM can have this non-covalent weak interaction. And in vitro, Rosen lab have shown that they can interact and drive phase separation to form liquid condensates, meaning that the interaction between sumo is strong enough to lead to phase separation, but weak enough they maintain liquid. So we think that the sumo interactions may be what's driving the APB condensation. More specifically, what we think is that after DNA damage response on the telomere, which can trigger by a shortened telomere, that leads to the modification of proteins on the telomere by stimulation, and then that will recruit proteins containing sumo SIM, including PML protein, and which has 3 sumo sites and 1 SIM. And this local concentration of sumo SIM containing proteins will phase separate because of the sumo SIM interactions, and leads to the formation of APB, where PML body is the dominant component, and then leads to clustering and enrichment in repair factors to elongate telomeres. So, and to test this hypothesis, we wanted to control the SIM enrichment on the telomere instead. The idea was if you have SIM, then that will kind of recruit the sumo in the cell to start the whole process. And so we fuse SIM to the EDHFR, and this is chemical dimerizer. We fuse the halo to a telomere binding protein TF1, so that the halo now is anchored on the telomere and dimerizer as well. So when we turn on the dimerization, and you can see this is before dimerization, you can see the SIM is diffusively localized, and you see the telomeres, and which is shown in green in the composite picture. And you can see now after we turn on the dimerization, and SIM is recruited, but it also leads to morphological change in both channels, meaning that the foci become rounder, they become brighter. And we can quantify this process by looking at the images and then quantify the foci intensity, integrated intensity, which represents the size and the brightness of the foci. You see they both increase. And also you can see that even the telomere channels start to increase. And this is because when SIM undergo phase separation, the TF1 that is dimerized to the SIM also undergoes phase separation. Another reason the foci are increasing in brightness and size is because the number of foci is decreasing, and meaning that they are fusing together as shown in the movie. And this fusion event highlighted in here, you can see they become round. And because they label the telomere, so also it's a telomere clustering, which I will show with like fish to demonstrate that. And dynamic exchange of components with FRAP. And if we recruit a SIM mutant that cannot interact sumo, and we can see the dimerization still work, and that meaning that the SIM mutant can be recruited to the telomeres. However, we don't see phase separation, signature of phase separation, that the roundness and the change in the number, size of the foci, which I'm not showing you the quantification due to time limit, but we have those data. And then we next look at whether this recruitment actually induced APB formation, where we used telomere DNA fish to look at the telomere DNA, and we use PML immunostaining for the PML body. And this is with our recruiting SIM, which means the endogenous odd cells. And you can see the colocalization, the white one as the endogenous APB. And if we recruit SIM, we can see that there's a dramatic increase in the number of colocalized PML body on the telomeres, meaning that APBs. And this is not saying if we just recruit the SIM mutant that I just showed you, and the quantified here that both the number and the intensity of the APBs increased after we recruit SIM. So this agrees with our hypothesis where the simulation process will trigger the phase separation and leads to formation of APBs and the telomere clustering. Next, we want to ask, are the new APBs formed that we show with immunostaining actually by the de novo formation of a nucleation of PMO bodies on the telomeres, like we hypothesized here, where the simulated telomere will recruit those proteins and leads to phase separation. So to answer this question, what we did is we now don't label the telomere channel. Instead, we label the PMO protein with the green channel. And now we recruit some. You can see now some folds have started increase because now they go to the telomeres. But we don't see too much change in the number of PMO body that. And also, if we just look at the colocalization of PMO body on telomeres after the dimerization, we only see this kind of fusion event where some enriched telomere will fuse with the existing PMO body, and we never see new PMO body form. So we wonder why, and that is what is wrong with a hypothesis. We thought that might has to do with whether the PMO protein is simulated or not when they're not in the PMO body. So this is the nucleus, the PMO body. We know that PMO body is a hotspot for simulation, so that the PMO protein is modified by SUMO, 3 SUMO site, 1 SIM. But outside, the simulation may be lost because simulation is always locally controlled. So in this case, if we recruit SIM on the telomeres, then the telomere will be able to enrich SUMO, but it will not directly recruit proteins contain SIM only that does not have SUMO. So in this case, then we would have SIM enriched droplets where the proteins contain SIM, the SUMO, the endogenous SUMO and SIM can phase separate, leads to phase separation, and that will fuse with PMO body to form APBs. So if this hypothesis is true, then if we just directly recruit SUMO to the telomeres, now we have a SUMO enriched telomere, then that will be able to recruit the proteins, the PMO SIM containing protein, and then leads to the phase separation and formation of PMO body on the telomere. So we tried that, and now recruiting SUMO 1, we also tried SUMO 2, SUMO 3, see similar result, where we do see that if we recruit SUMO, we see no new APBs and PMO bodies are formed. You can see the number start to increase. And then we can show that as shown here in this one, and we can see that a SUMO enriched telomere will gradually grow a PMO body on that telomere. So we can quantify this, and we see the majority of the APBs are formed with nucleation in this case. They're still fusion, meaning that there are still simulated telomeres can fuse with existing PMO bodies to form APBs. So they're actually the two pathways for the APB formation. Both can coexist: de novo formation, de novo nucleation, or fusion with the existing PMO body. So because if APBs can be formed by a non-APB and fuse with a PMO body, meaning that there must be some phase separation happening here. And for a SUMO enriched telomere, it must recruit some other proteins other than PMO protein to drive this phase separation. And we think those might be those DNA damage proteins that are shown and to contain SUMO SIM. And we have data to show that indeed, if we just recruit SUMO, we can see enrichment of some of those proteins, including BML, which is a helicase, and RAF52, RAF80AB1. Those are proteins that are involved in alt pathway being enriched after we recruit SUMO. And so this would indicate that if we have a non-APP condensate formed on the telomere and we have APB, there may be multiple ways that telomeres could cluster together. In addition to the APB clustering that we hypothesized, there would be APB clustering with a non-APB condensate, and there will be fusion between a non-APB and non-APB condensate on telomere that leads to telomere clustering. To test this, whether the the PML that is required for the clustering. In other words, whether non-APB involved clustering exists in those cells. We're trying to look at telomere clustering in PML deletion cell lines, alt cell lines. So we use this assay our collaborator has established, where we create DNA damage on the telomeres, use nuclease FOC1 and fuse to the telomere protein GF1. And then we use live imaging to look at whether telomeres can still cluster. And here we can see that indeed they cluster fine. And if we quantify actually the number of telomeres, which reduce the degree of clustering, we don't see a huge difference between the wild type and the PML knockout cells, indicating that PML doesn't play a significant role in this case. But we're using FOC1 fused to a telomere binding protein that constantly cutting the telomere DNA and generating DNA damage response on every telomere. We wonder, is this true for endogenous pathway where we don't have this persistent DNA damage and not every telomere is damaged, so they can like find each other and fuse? So to do so, we then synchronize cells in the G2 phase. That's where the endogenous homology-directed DNA repair happen on the telomere. And then we look at the enrichment of SUMO and also the telomere clusters. And we see that if you look at the arrow indicates the number of SUMO that enriched on the telomere DNA, we see a reduction of the SUMO in PML deletion cell lines. And we also see the telomere clusters actually induced. You can see the large blobs indicate telomere cluster. As quantified here, the clustering is reduced, and then the SUMO is reduced. This means that PML although it's not required, it really helps efficiency in endogenous cells in two ways. First, the presence of the PML protein can promote stimulation-mediated phase separation because it contains 3 SUMO and 1 SIM, and PML protein can self-interact, drive phase separation. So there's another driving force for protein interaction. And the existing of PML body, that the ability to fuse with a stimulated telomere, they facilitate the efficiency of telomere clustering, which is important because the cells have to undergo the telomere maintenance in that G2 before they go to the next cycle of mitosis. So far I've shown you that the formation of APB and telomere clustering has to do with stimulation-mediated phase separation, also the recruitment of DNA repair factors on the telomere. But there are also many other abnormal features that I did not mention. One is the existence of the extra chromosomal telomere repeats in those cancer cells that is used as a marker. The other one is the overexpression of TERA, which is a telomere repeat containing RNA that is transcribed for telomeres. And so we're asking what are those, how those phenotypes are originated in those cells, and what are the consequences. And for example, we're asking if the telomere clustered, does that affect the actually chromosome organization, right? And then what's the consequences of that? We're using tools to artificially induce clustering and using Hi-C to look at the chromosome contact, for example. And we're looking for postdocs if you find any of the projects interesting. So, last but not the least, acknowledge that most of the work that I present today is done by my graduate student Ronwei and collaborator, and particularly the dimerization system is developed by the Sherman Wave Lab at Penn, and we continue to collaborate. And the funding from the 40M for supporting our work. And thank you for your attention. Happy to take any questions.
So I was curious about the fact, are you able to overexpress or basically play with the concentration of your different molecules? So playing with SUMO or playing with a particle of the PML body in order to also see if this somehow respects the standard liquid phase separation properties that we expect from when we play with the concentration, for example.
That's a good question. We one of the reasons we don't want to directly recruit PMO protein is we know that overexpressing PMO protein is not good. You have like huge PMO bodies and just like so many artifacts that is so that's one of the reasons we want to go to like actually one step earlier to try to mimic this recruitment, so we don't change the PMO level. And even the PMO live imaging that we show is a CRISPR cell line with PMO labeled. We do believe, though, we have tried overexpressing PMO protein to see what we get. We just see increased number, more efficiency in telomere clustering. So we think the number matters, and PMO body is really helping sticking telomeres together if there are a lot around. And we some of them we overexpress sumo. The original idea was not to overexpress sumo, but since we see a difference, now we have to overexpress sumo. But overexpressing sumo, of course, does have its side effects. But we don't see we see the size changes, not necessarily the number of things that we look at. So I guess the reasoning would be because PMO protein is able to self-associate, and then sumo has to be conjugated. So the overexpressed sumo can be absorbed by the cells more.
Okay, and maybe a second question regarding the coarsening dynamics of your bodies.
Mm-hmm.
So it's quite. I mean, since these bodies are attached to telomeres, so basically they are constrained also by the polymer. Okay, the fact that you are attached to something. So can you see some functional effect of this slow motion, more or less, of this slow coarsening of these bodies?
Yes, we're trying to look at that. So like my student Tafazwa, she's looking into that. So we definitely see a slowdown, and like the one thing we observe is you will observe more clustering events within like 2 hour, 3 hour window. And if you follow them for longer, the plateau, the clustering become less efficient. And we think just because now they are more stuck, and they have to drag multiple chromosomes around, and there's no way that they can do that efficiently. And so there's definitely that. But we think once they're close enough, though, which is another thing interesting we observe, but we haven't followed up, is there's a difference between if you just have a sumulated telomere fused with a sumulated telomere, and they look different under the microscope compared to when you have like PMO body fused with telomere. And we think probably because the existing PMO body has a structure, and that will lead to the difference in the kinetics, and maybe even the structure will look different depending on whether the PMO body is growing on the telomere versus that it's like fusion with the PMO body. So we do see that the composition of the droplets on the telomere affects once they touch, but the kinetics of fusion is affected by the chromatin. We haven't found a good way to quantify that, though. In just like we see the number decrease over time, but we if you have a good idea like how to quantify that, I would love to hear your idea.
I don't know, because I mean, for sure, you may know that there is different. I mean, in standard phase separation, there is different coarsening dynamics. So there is Brownian motion, so fusion, as you were showing. There is also Oswald ripening. I don't know if you know that. So basically, that small droplets may evaporate and go into the large droplets. So I think for nucleoli formation, which is also associated with phase separation, it was shown that Oswald ripening was repressed somehow. So I was curious also if you can quantify that. I guess you cannot, but it's more like a physicist question.
Yes. So the ripening, which is interesting, we never see ripening when they're on the telomeres. But we've tried to use our dimerization system to induce droplets outside of the nucleus. We do see ripening. Like it's very obvious ripening where droplets, some of them are decreasing over time, and we're looking into it. And also we have some very preliminary data showing some proteins we see less ripening, some proteins we see more ripening. But the ones on the telomere, we never see ripening. We don't see a foci just like disappear. They either bump into another one or they persist. Probably
Okay.
because they're stuck on the telomere. So they're like, there's always something. Like the first layer, for example, well, because our system is artificially literally link them to the telomere. So they're always there. But the ones we've tried to induce droplets, not on any chromatin, but in the nuclear plasm. And we do observe ripening.
Okay, so that's interesting because that's some. I mean, we are working also on HP1, which also may have this capacity to phase separation, and we have the same. I mean, we observe the same thing, that basically when, of course, HP1 is localized around heterochromatin, you suppress this Oswald ripening effect, and you basically are stuck because of the very slow motion of the chromatin, and so coarsening by Brownian motion is very slow, and it's,
Yep.
and it's also
Similar happened to we've also tried to if we just recruit proteins to centromeres to form droplets, we see signature of phase separation because the protein, the background get depleted and more than just we don't have phase separation protein. There's barely any coarsening through fusion.
okay. Yeah.
I think maybe because the centromeres are just less mobile than the telomeres. And we also thought, we haven't looked into this with too much details, has to do with the odd cancer cells have a kind of maybe more mobile nuclear, something like that, where the telomeres maybe can move more. Or there's other people suggesting that if you have DNA damage, you trigger actin-based mobility to facilitate in the endogenous system. And those things we're looking into. But passive diffusion is sufficient enough to drive clustering. And we're looking at whether is the active process will increase efficiency. And the idea is, again, about efficiency, the cells have to do the job before the next mitosis. So they're not stuck in G2 to finish the repair. So we think that may happen.
So you were explaining that, if you tether SIM to TRF1, you get increased interaction with increased formation of APBs. But which protein on telomeres is actually normally involved in either is being stimulated or has SIM properties? This I missed.
That's a great question. So TF1, TF2 are being reported, being stimulated, and it's been shown that it's important as a signaling for triggering the APB formation. But honestly, though, I think in the pathway, probably the stimulation is more on the DNA repair protein that's being recruited to it, because you have lots of protein already probably being there when stimulation happens. So this kind of stimulation has this kind of group behavior where it's going to stimulate proteins around it. So we don't have evidence to say this, but I think it's not only TF1, TF2. TF1, TF2 happen to be stimulated. Is the regulation of the ligase then create a hotspot? And I think that's why you have stimulation everywhere on the chromatin, but PML bodies don't go there. I think it's about how much proteins is being modified. And because you have all those DNA repair proteins concentrated on the telomere, because telomere is repetitive, you simply increase the number enough. Now you can deplete PML protein from the existing PML body. But if you just have regular stimulation of a small protein, like, for example, involved in transcription, but you don't see PML protein go there, I think it's because you are not passing that threshold to drive a local phase separation yet.